are two different processes that when you inhibit them, it actually causes the DNA to open up more, and as we just learned, that results in more mRNA production from them, and thus more protein production. So what we're doing here is we're using our two drugs, decitabine, which is a DNA methyltransferase inhibitor, and SP939, which is our histone deacetylase inhibitor, to cause, again, the DNA to open up and thus more transcription and translation to happen. So those are the two key players there, and what we found is that when you introduce these drugs to human cancer cells, we actually see increase of transcription of certain regions of the genome called human endogenous retroviral elements, and specifically in the long terminal repeat 12C family. What's really special about these to know is that LTR12C activation results in the production of treatment-induced novel polyadenylated transcripts. If we can think of a nice acronym for those, we can call them TINPACs. So these are those novel sequences of RNA that I was referencing earlier that result in the production of those novel proteins. So our TINPACs to the novel proteins to then, which can be digested, so it's broken down into smaller protein units that we'll call peptides. And these peptides can be presented on the cancer cell surface. And here's the exciting part, which leads to immunogenic neoantigen presentation. And what that basically means is immunogenic means that it can be recognized by your T cells. So in this case, we're talking about CD8 plus T cells. And then neoantigen just refers to the fact that it's a protein being presented on the surface of a cancer cell. Specifically, we're using MHC class 1 machinery to do this, so this is just a special class of protein receptors that cells use to present molecules on their surface. So that's a nice background for us there. And again, to bring it all home, we're using decitabine and SP939 are our two drugs, resulting in the end result of neoantigen presentation. So I'll be giving a special focus to decitabine throughout the presentation just because that's the main drug being studied. All right, so I hope we're all feeling comfortable and excited. With that, we'll move on to the actual experiments themselves. So what was my goal over the summer? My goal was to create a mouse model acting as a preclinical model demonstrating that upon decitabine treatment, we see this in-mouse phenomena of MHC1-mediated antigen presentation. Because all the data I just shared with you guys a few minutes ago, we saw this happen in human cancer cells. However, before we move to, of course, in-mouse testing and eventually, of course, the end goal of creating an mRNA vaccine for cancer patients, we first have to check, does this whole process indeed happen in mouse cancer cells so that we can set ourselves up for future experiments? So that was the goal for my experiments there. And this is just outlining that we need to make sure that it works in mouse cells before we move on to actual mouse testing. All right, and here we have our treatment groups. So we'll break it down super simple. DMSO is our negative control, so don't need to worry about that too much, but important to have in our experiments. Decitabine is, again, our DNA methyltransferase inhibitor. SP939 is our histone deacetylase inhibitor. And then lastly, we had that combination treatment to investigate synergistic effects. So we're really going to be focusing on the decitabine and then also the combination treatment today. All right, and again, just as an overview, we're introducing decitabine to our mouse cancer cells, hoping we see induction of these special repeats in the genome, leading to the creation of those TINPACs, those RNA molecules, resulting in the production of those novel proteins presented on the cancer cell surface, making them recognizable to our T cells. Wonderful. And the first thing we wanted to do is validate the beginning of this pathway, because you guys can see there's a lot going on here. So we want to ask ourselves first, is this indeed happening? And if so, do we actually see induction of these long terminal repeats in our mouse cancer cells? And if so, do we also see production of these TINPACs? So that was a big question with that. And I just want to familiarize you guys with the two transcripts of interest that we'll be focusing on today. So as you can tell, our MMER-VK10D3, IAP, those are different than the ones that we're talking about in human cancer cells. That's just because in mice they have different repeats, so you can kind of think of these as the equivalent of our LTR12C repeats, except in mouse cancer cells. So these are the two repeats that we'll be focusing on. This data is from my mentor, Dr. Qurat-ul-Ain. He did an experiment earlier on in mouse cancer cells. Basically, he was looking for the right repeats for us to investigate once we got to my experiment. So that's what this graph is from, and the big takeaway here is that's going to be the two repeats that we're focusing on today that we're hoping we see overexpression of. So moving forward, again, TINPACs leading novel proteins to peptides, so won't forget it. All right, So here's the data from that first repeat and analyzing. So this is the culmination of my work from the summer. So I was treating the cells with cytidine. We isolated their RNA, and then we got to see, did we indeed see overexpression of these specific repeats in our mouse cancer cells. So this was really exciting because this shows, yes, for this one we did. So as you can see, with our decitabine and SV939 treatment, we indeed saw an increase in mRNA expression. So this is saying, fantastic, we're seeing production of these novel tin pads. Same thing with our MMERVK10D3 repeat. Again, our decitabine treatment and the combination treatment is resulting in a significant induction of these repeats in cancer cells. So this is really exciting for us because like I checked that second half of the pathway, because again, we've seen that this happens, but now are we indeed seeing these antigens being presented on those mouse cancer cells? And if so, you'll see we're going to get to map exactly where they came from in the genome. So let's do it. First, RNA sequencing, we used Illumina and Nanopore. Illumina, there are just two different ways to sequence RNA. Illumina is short read, Nanopore is long read. What I want you guys to know from this is that basically, where you could think of this as our DNA, and we're just looking for mRNA expression from certain regions of the DNA. So this allows us to map the mRNA we're seeing. Where in our DNA is it coming from? All right, and then immunopeptidomics is a really powerful tool to pair with this. We're going to get to isolate those proteins being presented on the cancer cell surface and analyze them. So really we are able to bring everything together. So this is the results from the immunopeptidomics experiments. This is from Dr. Jonas Becker at the German Cancer Research Center.